Well, I guess I'm just supposed to start talking. Hello, everybody. I have a lot to say, so I'm just going to jump right into it because I think I'm going to run right up into my time limit. My morning presentation today was all about the good, fun, happy news about how cool honeybees are, and that's not what today's presentation is about. This afternoon, we're going to be using sort of an aphorism or a general piece of advice that I give to everyone, which is never underestimate a parasite. We're going to be talking about virroa mites. We're going to talk about their history, their biology, the way that they're transmitted between the colonies, and we're going to talk about virulence. I'm going to do my very best to teach you some pretty high-level evolutionary biology here, and I'm going to use cartoons to do it. So I hope that everybody follows along. So this is not an acceptable strategy. You cannot, whether you are a treatment-free beekeeper or a treatment-resistant beekeeper or a new beekeeper or an old beekeeper or whatever you are, you cannot simply ignore virroa mites. You cannot simply say, I'm not going to know about or think about this problem because they're gross and they make me sad and I'd rather just focus on happy things like dance languages. We've got to be masters of the parasites in our colonies. And so I will tell you that as a Boy Scout, one of the things that I really enjoyed doing was going out and hiking, going camping, going to beautiful places around the country and around the world and getting to experience them. And if people have that same mindset of, oh no, we don't want to deal with these parasites, they might have just sent me out and said, good luck, hope you don't get Lyme disease from all those tips. But that's not what we did. We went out, we didn't cancel all of the trips either. We didn't say we're not going camping anymore because Lyme disease exists. We said, here's how deer ticks work, here's how Lyme disease works, here's what you can do to protect yourself, here are some tools, here are some procedures we're going to use that will keep you in good health. And then we were able to go on the camping trip. So the same thing is true of virroa. We need to know our enemy. We need to, as Sun Tzu told us, we need to understand our enemy and ourselves if we are going to be able to fight them. So why should we study virroa mites? What's valuable in studying? Well, obviously we want to be able to kill them, right? We want to eliminate virroa. But I ask you what you actually know about this animal, not how they affect your bees, but what do you know about the virroa mites themselves? And I hope harmful in this, we're really using something that in science we call virulence. So a more harmful disease, a disease that does more damage to the host, we can think of as a virulent disease. And so the question is virulence going to go up or down? So this is a little cartoon schematic here that illustrates the thought experiment we're going to do. We've got two organisms. We've got this red, pointy, angry parasite. We've got this big, sad, blue host. And we're going to assign some values to the parasite. Really, we're going to assign some values to the system. And these numbers don't mean anything. I just pulled them out of thin air. But we can use them in comparison to one another. So we've got two values. We're going to give a reproduction value to the parasite. How quickly does it reproduce? How quickly does it spread? How reproductive is it? And we're also going to have a host death measurement. And that host death measurement is going to tell you what percentage of the infected hosts are going to die. So in this case, in our example, we've got reproduction of 3, host death of 10%, right? Especially early in COVID, these numbers were pretty reasonable, right? This was sort of the situation we were looking at. And the question everyone had was, what's going to happen here? Is it going to evolve so that it spreads more easily? Is it going to evolve so that it kills fewer hosts? Or is it going to do the opposite? And so when we try to make these predictions, one of the things we can do is we can look around at other examples. Not identical examples, but maybe similar ones. So if we look at another system, maybe we've got another parasite and another host, where we see really, really high reproduction. The parasite spreads really easily, but the host death is very, very low. Hosts dying of this is very uncommon. Maybe that's something like the common cold that we would compare to something scary like COVID, right? So in that example, now we've got two comparisons, and we might sort of wipe some sweat off our brow and say, good, we can expect that the parasite in the host system is going to go in that direction if we just weed it out. But unfortunately, that's not really how this works. So if we have a time machine, we could actually go into the past and look at a whole bunch of different parasites and hosts that were similar to the one we're trying to make predictions about. This might be what we find. We find that maybe in one example, the parasite reproduces really slowly, but it kills a lot of hosts. In another example, maybe the parasite reproduces really fast, and it hardly ever kills hosts. In the third example, maybe it reproduces really quickly, and it kills a huge number of hosts. So that's really the nightmare scenario for a disease, right? It's spreading very, 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 very quickly, and it's killing almost everyone that it infects. So if we then jump to the future, and we're saying, all right, good, well, based on how those things worked in the past, what do we expect to see with our new parasite host system? The problem is, we're not going to see all of those parasites and hosts in the future, are we? Because if you are in that first example, the parasite reproduces slowly, but it kills a lot of hosts, well, it might kill all of the hosts in the local area, and then before it's able to spread, all of those hosts will be dead, and so will the parasite. So now the parasite's gone extinct, and the rest of the hosts are fine, because they never got sick in the first place. It spread too slowly. In the next example, sure, maybe both of those things are going to emerge. In the third example, where the parasite reproduces really, really quickly and it kills lots and lots of hosts, it might lead to the extinction of the parasite. And how has evolution solved the problem in the past? Have these diseases gotten milder? We have to remember that there are ghosts that we can't see. Parasites. So this is abstract, but it illustrates the phenomenon we're dealing with our mites here, which is that we can easily trick ourselves and feel like the parasites in the host should get easier to deal with over time, even though that's not necessarily how biology works. The other thing is that if you were a parasite, and let's jump to bees now. Let's use bees as our example. So we've got one host, let's say that's our beehive. We've got two parasites. Parasite A and parasite B. Maybe one of them is a virroa mite that doesn't reproduce very much, or maybe one of them is a virroa transmitted virus that doesn't reproduce very much. One of these reproduces really quickly, it kills half of the hosts. One of them reproduces much more slowly, and it doesn't kill a lot of the hosts. Now, as a beekeeper, which of these would you like to have in your hive? B, the second one, right? If you were, the bee colony that was infected by the two of these, which of them are you more likely to spread to your neighbors before you eventually succumb? The one that spreads faster, right? Which means that you're going to wind up with the worst parasite here. The parasite that multiplies faster and spreads is the mean one. Nice parasites do not compete very well against mean ones unless they get particular advantages. So in general, the conclusion here is that parasites don't just naturally become less harmful. We can't just assume that a disease will go away or get easier or sort itself out. It doesn't happen automatically. If it's really, really easy for a nasty parasite to spread, if it's really, really easy for the bad version of it to spread, then the nice version is never going to have a competitive advantage. So the amount of spread between different hosts is a really, really important thing for us to understand, which is going to be true for a lot of these cases, in particular honeybees and varroa and viruses. It also matters how sick the host gets, because the host's sickness is going to be determined partially by what the parasite does, but partially by what the host does. You can have a really nasty parasite infect a resistant host, and the resistant host will be just fine. So when we talk about these examples of these parasites, these hosts competing with each other, we've been talking a lot about the parasites, but we've got to remember that the hosts can also, grow a thicker skin. They can evolve some defenses that will actually help them out. Parasites and hosts are an endless arms race. This is the key feature that we need to understand here. To it in our bees. But this arms race is the way that all of these systems get sorted out by nature, all the time, and it's happened for a very long time. So what do I mean when I say an arms race? Well, we can take an example like this. Two organisms that are interacting with each other in the environment, right? Well, this is working really nicely. It's a nice outcome for both of them, but there is a way that one of them, with a mutation, could get an advantage. It could decide that it's going to become a parasite. So this is a situation that might play out for a while, but eventually what's going to happen is they're going to be engaging in that fight, and a counter adaptation is going to emerge. You're going to wind up with something that counters the initial parasitism, some resistance, let's say. So over time, this is going to continue to play out, and it's simply going to become more elaborate as the arms race continues. You just develop more and more and more arms. And eventually, you will find yourself in a situation where you find heavily armored, heavily weaponed organisms interacting with one another, and more or less achieving some level of compromise, right? As long as neither of them gets too much of the upper hand, neither will go extinct. So if we want to look at an example like that, we might wonder what the history of the relationship between virroa mites and honeybees really is. And to do that, we have to zoom out a little bit and go back in time to 1916. Who can tell me what the red line is? Who can tell me what the image is? Right? The Trans-Siberian Railroad. That's exactly right. So the Trans-Siberian Railroad was completed in 1916. Other things happened in Russia in 1916. We don't have time to get into all of that, but the completion of that Trans-Siberian Railroad meant that czars slash, dictators of the USSR were able to move beekeepers from Western Russia to Eastern Russia. They could take beekeepers in Moscow and load them and their bees and their queens onto trains and send them out to Vladivostok. And they did that because there were beekeepers in Eastern Russia. There were beekeepers, and if you look closely, Vladivostok is just north of North Korea. It's just north of the Korean Peninsula. There were beekeepers there, and they were keeping bees. And those beekeepers weren't making a lot of honey, and the bees swarmed a lot, they absconded a lot, they weren't terribly cooperative, they didn't grow into big colonies that made a lot of wax and honey. And so they said, well, why don't we go out and hybridize some of our good Western bees with some of those crummy Eastern bees? Well, the problem, unfortunately, is that, sure, we've got these two different bees here, but they weren't just two different races of bees, they weren't just two different varieties, they were actually two different species of bee. We had Apis mellifera on top and Apis cerana down below. Apis be kept in hives, and they traditionally are in a lot of Asia, but they are not as productive in a lot of different ways. So any hybridization effort, any crossbreeding was doomed to failure. And eventually we know that some of those beekeepers that were transported decided that this whole thing was a bust, and they wound up coming back to Western Russia. Obviously, behind the Iron Curtain, it's hard to get really definitive data about this, but we do. 2009. Somewhere between 2010 and 2012, they showed up in Madagascar. They came in probably on a cargo ship or on an airplane. And I actually spent some time, this is me and one of the field technicians in Madagascar, after my PhD, before I got this job at Betterbee. I spent 3 months living there and working, trying to identify virroa resistance traits in their native honeybee species. But unfortunately, they are just as susceptible to virroa as our bees are, as any bee was before any selection started taking place. And so this is a catastrophic, devastating threat. And so we need to understand what it is that they do and why they cause this harm to our bees. So if we look at this organism here, what can we say about it? It looks scary. It's got 8 legs, right? Not 6. So it's an arachnid. It's more closely related to mites and ticks and spiders and scorpions than it is to our honeybees. That gives us an advantage. It means that if I want to use a chemical that is poisonous to arachnids but not poisonous to insects, I can. That's where these miticides, these synthetic miticides, come from. It's using its 2 middle legs as almost like antennae. It has sensory hairs that allow it to smell the air around it, and it waves them around because it's not an insect. It doesn't have any antennae to sniff the world, but they can smell what's going on around them with those legs. If we look in between those legs, we can see its sort of elongated mouth parts, its chelicerae. Those are actually going to be wedged through the exoskeleton of our bees so that they can feed on them. What you'll also notice is if we zoom in on the head of the virroa mite, there's not very much head there. Virroa mites don't have a lot of food, and so they've gotten rid of extraneous things that they don't need. One of the things they've gotten rid of is their eyes. They're completely blind. So we are fighting a blind threat to our bees that have different, unique physiology compared to insects, and that have been specialized on parasitizing honeybees for a very, very long time, just not our species of honeybees. The virroa mite, this is a less scary photo when you don't have them in black and white. It looks slightly less like Godzilla, but they're still enormous compared to our bees. We think of them as, they're little, they're parasites, they're like ticks. I don't like having a tick on me. Well, your bee doesn't like having a tick on her, but this is a heck of a lot bigger than a tick is to you. This is the size of a blood-sucking dinner plate crawling around on your back, right? There are two different modes that virroa mites have that they alternate between. They can be in the reproductive mode, where they're going into cells, feeding on the young bees, laying eggs, and having more offspring. And they can be in what we call either the phoretic mode, or it's sometimes called the dispersal mode, where they're riding around on adult bees, moving between them, feeding on them, and then waiting for their next opportunity to go into a cell and reproduce. We know that it's very difficult to study the mites, but we estimate, I think this is mostly work that Marla has done, her lab has done, we think that the mites go through about 3 reproductive cycles over the course of their lives. So they're going to alternate between reproducing, being phoretic, reproducing, being phoretic, reproducing, being phoretic, and then they'll die somewhere in that process. The reproductive mode is pretty straightforward. The mite goes into the cell. She runs into a cell that's just about ready to be capped, and then she hides herself down in the brood food. If we can find a way to poison that brood food, wouldn't that be a neat way to fight virroa mites? But if you don't study the biology, you're never going to find those weak points. After the cell is capped, the mite crawls out, she starts feeding on the bee, and then she's going to lay a series of eggs. She's going to lay one unfertilized egg that grows into a male, because even though they're not very closely related at all, honeybees and virroa use the same sex determination system. Unfertilized male, fertilized female. She lays one unfertilized egg that grows into a male, and then a series of fertilized eggs at 30-hour intervals that will grow up into females. All of those young mites will feed on the bee. The mother mite, as she's feeding, is going to go up to the top of the cell. She's going to poop at the top of that cell and create a little pile of feces. And as her offspring feed, they're also going to go up and they're going to poop in that same spot. That's important because these are blind animals that navigate by touch and by smell. So how does the male mite find his sisters to incestuously impregnate them before this bee finishes maturing? Well, they go find the pile of feces that their mother started, and that's where they have their rendezvous. So it is a grotesque biology, but it's one that we need to understand if we're going to fight them. The male virroa mite cannot survive outside of the cell. He's much weaker. He's used to being in a very, very controlled environment. That means that when you use something like formic pro, when you're killing your mites by having formic acid penetrate the cell, they may not kill all of the female mites that are in there, but they might kill that weak male mite. And that male mite might be unable to impregnate his sisters, and now you've got a bunch of infertile mites running around. Not so bad. This is what that mite feces looks like. These little white residues that you can see if you tip a frame up are called guanine, which is just what we call the poop of mites, spiders, ticks, arachnids. So that guanine residue can give you a signal of whether you've got a little bit of mite reproduction or a whole lot of mite reproduction happening in your hive. The other thing that you can examine is mites in their phoretic or their dispersal mode. All of the mite measurements that most of us are doing, with the exception of measurements where we go and actually open up cells and pull out pupae, are going to be focused on identifying how many phoretic or dispersal stage mites there are in the colony at a time. It doesn't tell us what's in the brood. So if we look at this photo, this is maybe my favorite photo that I took in graduate school because it illustrates something really important. This was a colony where I was breeding as many virroa mites as I possibly could. And in this colony, I had, it was an observation hive. And what you can see is that I've circled a whole bunch of bees that have not just one, but more than one virroa mites on their bodies. What's interesting is not that the virroa mites are riding around on the bees, that you can see them through the glass wall of the hive. What's interesting is that we can see the undersides of just a few bees, and we can see that they're covered in mites. We can see the backs of a whole bunch of other bees, and we can't see a single virroa mite on them. Do you think that the mites were only on the bees that were walking around on the grass? No, they were not. The mites were on all of those bees. But because the mites are smart, they're going where they need to go to feed on those bees and to avoid being groomed off by the bees themselves, they are going to go to these areas on the underside of the abdomen. That means that if I pull out a frame and I look at my frame and say, I don't see any mites, so I must not have a mite problem, I'm deluding myself. Unless I'm sitting there and picking up every bee and flipping her over and looking for mites, you're not going to notice it. So in Apis cerana, the original host species. Yes?
Is that photo on like the brood?
Yeah, this was over the brood nest. It's a complicated experiment. We actually just published it. So it was an observation hive that was dying going into the fall, so there wasn't much food left.
Where did you publish that?
I think it came out in Apodology, and I'm not sure if it's in the most recent issue or if it's just online. The citation is Smith. So in Apocyrana, the original host species, we've got mites only infesting drone brood, and multiply infested cells actually get entombed by the bees. If two mites go into the same cell, have our bees and these mites interacting with one another. Less than 100 years, which is about how long we think these mites have been around bees, is simply not enough time for an evolutionary equilibrium to emerge. If they did reach a compromise, it might not be a compromise that we like. Remember that example of the different kinds of hosts and parasites, and how sometimes the parasite just makes the host go extinct? Well, if that's the equilibrium nature is taking us towards, we need to do something else, because otherwise we lose our bees. And if that wasn't bad enough, we also, of course, know that these mites are vectors of a great number of bee viruses. This is a list of bee viruses on the left. Many of them we know can be transmitted by the mites. In particular, deformed wing virus, virroa destructor virus 1, which is a cousin of deformed wing virus, and a number of these others, these paralysis viruses and things, can be transmitted by virroa mites to honeybees. And so this is just one photograph that illustrates a bee who has grown up with a large amount of deformed wing virus and has therefore failed to properly inflate her wings. She's not the only bee in the colony with deformed wing virus. She's just a bee who had so much in her body when she pupated that she wound up showing the sign of the disease. But the rest of her colony probably has high levels as well. Sure. Yep. Oh, it's not going to hurt the queen.
No, how is it for this bee though? Because all the eggs are already in the bee.
Oh, because the virus is transmitted directly by the mites themselves. When the mites are feeding on the bee inside that reproductive cell as they're reproducing, when their mouth parts are sucking nutrients out of the bee, they're also regurgitating virus into the bee. Thank you. So, if we do a very rough model of how the bee population and the mite population changes in a colony over the course of a year, it might look something like this. We start in January, the yellow line is going to show us our bee population, and that bee population is going to be low. It's going to increase dramatically in the spring until it reaches its peak. Then it'll be held for a while and then start to decline in the fall as they get ready for winter. And then we'll lose some of those field bees and we'll be left with the nice tight winter cluster. During that same period, the red line shows us the mite population. Hopefully it starts low at the beginning of the year, but every time those bees are producing brood, that's an opportunity for the mite population to increase. And the mite population grows exponentially as they continue to reproduce brood. So we can see a mite population that climbs dramatically over the course of the year. It's going to lag a little bit behind the bee brood production, but it's tied very closely to it. So that when we get to the end of the season, we've got a whole lot of virroa mites on not that many bees. Now that's not good. That's a bad situation to be in. And for a long time, people thought, well, obviously what we need to do is buy miticide and treat our mites just once around maybe August, September. We'll just knock this catastrophically high mite population down to 0, and then everything will be fine going into winter. Well, the problem is, even if you kill every one of those mites, you haven't eliminated the fact that you had viruses being transmitted and amplified in that colony over the course of the entire year. Every single time a mite reproduces, every single time that line goes a little bit higher, it represents mites feeding on bees. And those mites feeding on bees are having opportunities to transmit viruses to them and between them. And so this means that even if we knock that mite level down in September, we've still got all of this accumulated virus feeding and transmission, mite feeding and virus transmission that is taking place. So your goal as a sustainable mite manager needs to be year-round mite control. You want dead mites just before the winter, but you also want low mites throughout the rest of the year, because dead mites right before winter is not going to keep your bees safe all winter. If your mites survive your treatments, or if mites are able to invade your colony from somewhere else, you can't pat yourself on the back. It's still your problem. It's still you. And if you keep your mites year round, it is the easiest thing can't sit on a slide that says, oh my God. But in each file, it's going to be the meltdowns for your piece. So what I want to talk about next is this idea of transmission, the movement of mites. And so we've already talked about this spread from a sick host to a healthy one. Then we're going to evolve nicer parasites, right? The easier it is for the nasty parasite, the likely it is to make a host sick, and then jump to a new host before that other dies. So how might spread? Well, with colonies, not very something like that, right? It's simply about these mites on them. The mite decides to jump onto another bee, then onto the comb, and jumps onto a bee. That's not terribly interesting. What's a lot more interesting is how you can wind up with a colony that has mites and a colony that has no mites for you, and then wind up with a whole bunch showing up in the nested colony. So there are a few ways that this can happen. Something that you can call vertical parasite. That means that it is transmitted sort of down. In this case, that means that I've got a sick one that swarms. The swarm moves into this empty box. Now I have two colonies, both of which have related to each other. This would not be a terrible situation, because if that was, it would mean that a mite that killed its host would be to get into mutilation. They eventually go extinct, and we would help perfectly. Unfortunately, this is not the main mechanism. There's also horizontal transmission, where a sick host can get a healthy host sick as well. So in this case, we've got two occupied spots. Asking how the mites get into the hive. One is this. So one way to make this. Another mechanism is through robbing. A healthy colony and a sick colony next to it. And the healthy colony will say, hey, let's just go some time. And when they do, they'll come home to so there's mechanisms that can allow mite transmission to take place. If you want to diagnose robbing, kinds of robbing. So here you can see obvious robbing. This is the kind of or you look out towards the yard, you go, oh, something's up. There's going to be a bees. This might be that. So when you look but there's also overtop. So this is like experiment. And what we had here is very, very light colored yellow bees. But in an apiary that had dark colored bees that were very, very dark abdomens. And if you look closely here, this is a repeating animation. You'll see a dark colored bee and a light colored guard fighting one another. Eventually the guard sort of grabs like the but right in this also see that there is some degree of taking place in what looks like otherwise almost normal colony. So you can be robbing even if you don't see it. This is the aftermath of that experience. The colony the next day came back, found the yellow bees, the guard, but Robert had given up for a while.